All right, thank you everybody. Thank you to the organizers of today's symposium and to everyone here at OHBM. I'm Jacob Miller, and today I'm going to tell you about a new anatomical landmark in human prefrontal cortex that's important for anatomical and functional gradients. The prefrontal cortex is highly studied for its intricate role in higher order cognition. However, the modern templates we all use for the prefrontal cortex are lacking anatomical structures. So prefrontal sulci, when we average brains together into templates, shrink. This is shown on the bottom row here on an average cortical surface from 20 subjects, and here sulci are in dark gray with gyri in light gray. As we add hundreds and even more hundreds of subjects to these average templates, sulci in the prefrontal cortex shrink very small and disappear almost entirely. So this can be seen in between the superior and inferior frontal sulcus. As we include up to 700 subjects, these sulci almost disappear. This is in contrast to in individual subjects. So on the top row here are 3 inflated cortical surfaces from individual subjects, and in between the superior and inferior frontal sulcus in this case, these white arrows point out numerous sulcal indentations that are highly prominent within individual subjects, but are lacking from the average templates we use to study the prefrontal cortex. So we sought to go about characterizing these missing middle frontal sulci from average templates and characterize them within individual brains. And it's not surprising that these structures are missing from average templates, given that they've been historically mischaracterized. So in the human frontal lobe, there are 2 large primary sulci. There's the superior frontal sulcus and inferior frontal sulcus. In between them is the middle frontal gyrus, or MFG. Within the MFG, there are actually numerous smaller sulcal indentations, but these have been historically debated. Eberstaller in 1890 first defined the middle frontal sulci as the vertical and horizontal components, shown here in blue, but in the frontopolar region of the prefrontal cortex. Other anatomists continued Eberstaller's lead and left more posterior sulcal indentations in the middle frontal gyrus completely unlabeled. More recently, anatomists have begun to recognize that these posterior sulcal components within the middle frontal gyrus are in fact the middle frontal sulcus. And the newest templates of sulci in the human frontal lobe actually split this middle frontal sulcus into 3 distinct components. However, there's actually almost no empirical data as to how variable these sulci are within individual subjects, or even whether they exist at all. So we sought to go about characterizing this anatomical structure within individuals and see if it had both anatomical and functional meaning. So how did we do that? First, we went about manually labeling and characterizing sulci in the individual hemispheres of subjects. So we labeled almost all the sulci in the prefrontal cortex within individuals. Then, we used multimodal imaging data from the Human Connectome Project to go about quantifying the sulcal morphology, anatomical, and functional gradients within these individual subjects. And I'll show you using that data how the middle frontal sulcus is a prominent landmark within individual subjects, that it marks boundaries for gradients of both myelination and functional connectivity. So how did we go about manually characterizing these sulci in the prefrontal cortex? We started with the most recent and comprehensive atlas, or schematic, of sulci in the human frontal lobe, and this is from Petrides and Pandya. And we used this template as a way to guide our manual labeling of sulci within individual hemispheres. So to do this, here are shown 2 example left cortical hemispheres. We first identified the prominent central and precentral sulci in each frontal lobe, and from there did detailed manual outlining of the remaining sulci in the frontal cortex. We started first with the large primary sulci, shown here in blue, the superior frontal sulcus, and the inferior frontal sulcus. From there, we distinguished in the frontopolar region of the prefrontal cortex the intermediate frontal sulcus, shown here in green. Finally, we then manually outlined the remaining sulci within the middle frontal gyrus, here in white, as the middle frontal sulcus. In total, a dedicated team of undergraduates and I labeled over 800 sulci across 35 individuals in 2 hemispheres each. This Petrie schematic, however, conceptually separates the middle frontal sulci into 3 distinct components along an anterior to posterior gradient. When manually labeling, we also sought to determine if this structure could be split into 3 distinct sectors. And this was the case. Within every individual, the prominent middle frontal sulcus could be split into 3 components, an anterior, intermediate, and posterior component. Having performed this manual labeling, we then decided to quantify the sulcal morphology of these sulci within individual subjects, and I'll show you that data now. First, the middle frontal sulcus is highly prominent within individuals. Here on the left in blue, I'm showing the size of every sulcus in the frontal cortex, ordered by surface area. The middle frontal sulcus is shown here in dark blue, and each data point is an individual subject's hemisphere. The middle frontal sulcus is in fact highly prominent and actually has a comparable surface area to the primary inferior frontal sulcus. The 3 components of the middle frontal sulcus do not have any different size, however. So this sulcus is highly prominent within individuals, but it's also characteristically shallow. That's what I'm now showing on the right here in green. Again, this is ordered by sulcal depth, where higher values here indicate higher sulcal depth, and this is a normalized measure. Here in dark green is the middle frontal sulcus, and it's much shallower than any other sulci in the prefrontal cortex. Because there is a general relationship between sulcal depth and the time a sulcus forms during gestation, this points to the fact that the middle frontal sulcus is likely a tertiary sulcus that forms latest during development. Having shown that this sulcus is prominent within individuals, we then sought to determine if it could distinguish meaningful anatomical gradients. To do this, we turned to the T1 over T2 weighted ratio, pioneered by Glasser and Van Essen, and this serves as a proxy for myelination in the cerebral cortex and can be calculated within individuals. It turns out that the middle frontal sulcus indeed identifies a myelination gradient within the prefrontal cortex. To quantify a gradient, within each individual, for every sulcus in every hemisphere, we calculated its average geodesic distance along that individual's cortical surface from the central sulcus. This way we could relate myelin to the distance from the central sulcus within each hemisphere. And in fact, these 2 measures were highly correlated. Right now on the x-axis, I'm showing the geodesic distance from the central sulcus, and on the y-axis, this measure of myelination. So there's an overall relationship between distance from the central sulcus and myelination, and indeed, even the 3 components of the middle frontal sulcus are separable just based on myelin content alone. And that's shown in more detail here on the right. Again, each data point here is an individual subject's hemisphere, and myelination drops off from a posterior to anterior gradient, even within these 3 distinct components of the middle frontal sulcus. Finally, we wanted to see if this middle frontal sulcus could have any functional meaning, and to do that, we first turned to resting state functional connectivity. We used resting state functional connectivity network profiles calculated in individual subjects from a recent Thomas Yeo paper, which allowed us to calculate the overlap of each subject's middle frontal sulcal components with each of 17 Yeo resting state networks. That's what I'll be showing here, where this polar plot shows the overlap of each sulcal component with each of 17 networks. And indeed, the 3 components of the middle frontal sulcus show a distinct anterior-posterior gradient of functional connectivity, whereby the most anterior component of the middle frontal sulcus is highly overlapping with ventral attention networks, the posterior component is highly overlapping with default mode networks, and the intermediate component of the middle frontal sulcus contains a mixture of these 2 profiles. This was not just an artifact of averaging across individuals' profiles, however, and was indeed present within individual hemispheres. That's what I'm now showing on the left in 3 example individual left hemispheres. So these resting state network fingerprints were present within individual subjects as well. So in sum, the middle frontal sulcus is in fact a prominent anatomical landmark within individual subjects, and it marks boundaries for myelination and functional connectivity. And this is not a completely new idea, but rather perhaps confirms a theory proposed by Friedrich Sinides in 1964, where he theorized that tertiary sulci in association cortex in humans may serve as landmarks of transitions between both areas of cytoarchitecture and function. And so we're proposing that this middle frontal sulcus may in fact be one of those landmarks. Finally, to take advantage of the tedious manual labeling that we performed, we transformed all individuals' manual Sulcal labels onto an average template to make probability maps. And using these overlapping probability maps, we can then take unseen cortical surfaces within individual subjects, project probability maps onto their surfaces, and identify this middle frontal sulcal structure above and beyond current Sulcal labeling algorithms. And we plan to publish these as soon as we're done perfecting this algorithm and finishing our manual labeling. So I'd like to thank the entire Desposito lab, and especially Professor Kevin Weiner for teaching me how to find missing sulci. So please come by 2 posters from the Weiner lab where we'll talk about functional differences in these sulci across cognitive tasks and their relevance to behavior and development. So thank you very much.